Great. Good morning, everyone. My name is Tenthoff. I'm a senior biotech analyst at Piper Jaffray, and I want to thank you all for being here for the Cell and Gene Therapy Investor Day. Our next presenting company is Capricor, and Capricor is a cardiac cell therapy company conducting multiple studies. And I think what's really remarkable is the data that they just announced earlier this week from the HOPE Duchenne study for the treatment of boys with Duchenne muscular dystrophy. Capricor is also at the forefront of developing exosome technology. And here to discuss the story is President and CEO Linda Marban. Linda, we actually met at one of these events, I think 4 or 5 years ago. So it's nice to see both how the conference has evolved, how the company's evolved, and our friendships evolved. So it's really good to be up here with you. So I want to start, I know we only have about 13 minutes, and it's a lot of ground to cover, but I want to ask you maybe to start by telling us about the allogeneic cardiac cell therapy CAP-1002.
Thank you, Ted, and full disclosure, this is like one of my favorite people in the entire field, so I'm so delighted to be here with you today, Ted. We're a publicly traded company, so the typical forward-looking statement. So I think this is really my first time looking at an audience since presenting our Duchenne muscular dystrophy data in the HOPE clinical trial. And what I can say is that this has been one of the most exciting opportunities that we have embarked upon in our 10-year history as a company and in the last few years as a public company. What we have shown is that not only was the therapy safe and well tolerated, 75 million cells delivered down the coronary arteries, we showed really interesting signs of efficacy in the heart and also in the skeletal muscle, suggesting that infusing the cells into the heart delivered them through the body into the skeletal muscle and mediated a beneficial effect in the skeletal muscle. We also were lucky enough to see a peak response and the dropping off of a response so that we know how to redose in the next stage clinical trials. This sets us up for the next trial, potentially being a registration trial. We've already requested a Type B meeting with the Food and Drug Administration, and we're in the process of applying for both breakthrough and the RMAT, the 21st Century Cures Act designation that's similar to breakthrough. I think probably everybody in this room travels as much as I do. I say this is sort of like TSA pre-check, right? They give you, preferred entry, and you don't have to take off your shoes or your computer out of your bag. So we're excited to potentially have that. Shown here, I think, if I want you to walk away with one important takeaway message from this data presentation, is a dual responder analysis. So we showed that the patients had improvement in skeletal muscle function. 70% of the patients in our trial were non-ambulant. We call them fondly the HOPE-like patients. It's the HOPE clinical trial because they're a unique population of Duchenne patients in a clinical trial. They're older. The average age in our trial were 18 years of age, and 70% of them were non-ambulant. Actually, just by luck of dice, 80% of the treated patients were non-ambulant. So we were lucky that we had built in a measure of skeletal muscle function that didn't involve walking, and it's called the Performance of the Upper Limb, a validated test, one where the actual developers of this test are right down the road at AAN today, and they helped us build in this very important measure, which measures shoulder, elbow, and wrist and hand function. What we show here in a significant way in a very small trial, 25 patients, 13 got cells, 12 were followed with usual care, is that the patients that had an improvement in heart function, both in the reduction of scar in their heart and improvement in a contractile parameter of cardiac function, also had skeletal muscle improvement in terms of the elbow, which in this patient population is the most important measure. I will tell you that in showing this data to the thought leaders moving up towards data presentation, one of the advocacy people who is most well known in the space, Pat Furlong, said, "Linda, you realize that this means a boy might be able to hug his mother. And if that doesn't motivate you to get up in the morning and go to work, I don't know what does." So, one of the things I'd like to highlight is that we have an incredibly strong development team behind us. And we have an academic lab that generates incredible amounts of preclinical data. The lab is led by our founder, Eduardo Marban. And what they've discovered in the lab is that the mechanism of action of these cells is mediated by the release of exosomes. Exosomes are nanometer-sized lipid bilayer vesicles filled with RNAs and proteins that transmit a message of what a cell can be to what a cell could be. And the mechanism by which they work is scientific, which is called epigenetic modulation, which means it actually can change the behavior of a cell. Once we discovered the exosome, the world exploded for us, and it explained why in now 4 clinical programs we've been able to demonstrate a similar mechanism of action of these cells, which is a reduction in scar. And we also realized that you could then infuse the cells any blood vessel and get them to where they're going. So we did a proof of concept study where we actually injured a skeletal muscle in an animal, and then we infused in exosomes, and we saw them track to the site of injury and actually heal it. So basically, all you have to do is get the cells into the body, and they're going to find where to go and mediate their beneficial effect. I'll just close before we have a Q&A, because I'm always curious for what Ted is going to ask me, with the fact that we have a lot going on this year. We knew 2017 was going to be a big year for us, and in fact it is. We just announced the 6-month HOPE data. It's on our website if you want to listen to me speak again and actually see the data set of the top-line data. We're getting right in to share this data with the FDA and expect to initiate what we hope to be a registration trial of intravenous delivery, no more intracoronary, and repeat dosing in these boys and young men, because we saw the peak effect at 3 months. We also have an exciting opportunity in our adult programs, where J&J has been our development partner for the past few years. They have an exclusive license option on the cells for some cardiology indications. That option will be triggered by the delivery of data to them, which they'll be getting within the next month, and then they have 60 days to make their decision. And we always say make their decision, but let's remember that it's both companies' decision. We may decide to hold on to our cells so we can develop Duchenne ourselves. And then our exosome program, I always call that our butterfly, because it's in a cocoon. It's a caterpillar that crawled in, and it's making itself very beautiful. And we're going to be coming into the clinic hopefully in 2017 with potentially 2 exosome programs. Thank you.
Great. And Linda, I appreciate that overview. And I want to pick right back up with the DMD data, because that really, I think, steals the show. Now, in the HOPE study, in this 25-patient phase 2 study, the CAP 1002 was delivered via intracoronary catheter. And the goal really is, I believe, through this demonstration of systemic benefit of skeletal muscle benefit, to move more towards systemic delivery. So maybe you can kind of talk about the delivery in the phase 2 trial and what gives you confidence to move to systemic delivery in the phase 3.
Yeah, thank you, Ted. So while we were doing HOPE, as the exosome program was beginning to get legs, and we realized that you have to just get the cells into the blood to get the exosomes to the site of injury, we started a pretty complete preclinical development program in small and large animals, where we showed that we could deliver the cells intravenously, really simply in a mouse, for instance, putting it in the tail vein, and still see the same effect, both in the heart and in the skeletal muscle in Duchenne muscular dystrophy mice, the MDX mouse. And then, of course, we did some dosing studies in large animals to make sure that it would be very safe. We pushed the doses as incredibly high, higher than we'd ever pushed in a person. So we're very confident of not only the safety but the potential efficacy. Of course, we're going into the FDA with all of this data, and we think that we have every reason to believe that it should be. Certainly, I would rather have a catheter put into my arm than into my heart, although if it was life-saving, of course, you would absolutely put it in your heart. So, we're very excited at this chance to have more frequent dosing opportunities. We think, as I said, based on the clinical data, that we'll want to do redosing every few months. And so I'm sure the guys, especially the non-ambulant ones, were laying down in machines and on operating room tables, and it's a little challenging for them. We can just put it into a simple line and deliver them that way.
Yep. And you mentioned multiple dosing. So what do you kind of foresee as the dosing paradigm as you move forward, not just with the phase 3, but also assumably move into earlier stage and younger boys?
Yeah, so we're going to start looking at every 3-month dosing because that seems to be where we're seeing the peak effect. We're probably also going to try an every 6-month dosing. At this point, I don't see going beyond that just based on the clinical data. And what's beautiful, and I didn't get a chance to mention, is that the preclinical data has been very predictive of the clinical data, which doesn't always happen. And so, we feel like it's a good roadmap for us. Now, as we go younger, probably 2 times a year will be plenty, right? Once we sort of get them up to doses, the inflammatory effects can be calmed down, and we'll be able to hopefully back off so that they don't have to spend as much time, coming into a center. Although we anticipate the infusion will be under an hour.
Great. Excellent. And before I move on and kind of pick up with questioning on the exosome program, are there any questions from the audience on the DMD efforts and on that exciting data they presented? Well, again, congratulations on that. I think that's really remarkable for the field, and it's a great day for boys with DMD. So you, I think, very nicely showed that this mechanism of action is mediated through exosomes, and it's really work that you've been doing there that I think is kind of where Capricor is going to lean towards in the future. You mentioned that you may be filing your first IND this year, so maybe tell us a little bit higher level where these preclinical exosome programs stand. How broad could they ultimately be utilized? And maybe tell us a little bit about your first indication.
So let me just say that the discovery of these exosomes was the most disruptive thing that I think could happen in the field of cell therapy, because we actually have talked for years about the paracrine activity of cells. We kind of had this idea that something happened when a cell got in that caused a bigger effect than one cell, should be able to mediate. And so now when we understand that, these exosomes are released by cells, we now understand how cells work, and now we can move that to the next stage. You can collect the exosomes, concentrate them down, and even custom design them. So look for, in the next few years, people, Capricor and others, custom designing exosomes, packing them with really interesting things that mediate very significant and important biologic effects. For us, our Gen 1 product are the exosomes made by our cells. Our cells are potent anti-fibrotic, anti-inflammatory, pro-apoptotic, pro-angiogenic in their activity, and guess what? Our exosomes are the same. So this is really like being at a buffet. You can line up and look at all of the possible inflammatory diseases and say, wow, which one looks like it's the best? And we actually have seen some very interesting preclinical data, and I would say we have 3 programs that are in a neck and neck race into the clinic first. We got a grant with the University of Maryland from the National Institutes of Health to study hypoplastic left heart syndrome. We've been working on that program for a while, and we're going to bring this forward with our exosomes. We had originally thought about doing it with cells, but these are babies that are going to be getting this, and so why not give them a product which, even though the cells are completely safe when it comes to immunology, you still don't want to put them into a patient chronically that might get a transplant one of these days. And these babies could potentially end up being transplanted. So we're going to use the exosomes in them. It's a perfect place to start because we can inject them into the heart at the time of surgery. We also started off thinking that we would do ocular graft versus host disease. We thought we could only make small doses of exosomes for the clinic, and we wanted to do something where we could do local delivery. But the systemic delivery of the exosomes in a preclinical model of graft versus host has been so beautiful that we think that we're probably going to move forward with that as one of the major clinical indications, systemic graft versus host disease. And he's smiling because I asked his opinion on this before we move forward with it. And then we also have another cardiac indication that we're not talking about yet, but it's similar to some of the things that Audentes is developing, but it's not a gene therapy. It's using exosomes, and it fixes a certain type of cardiac disruptive type of electric problem. So that's the exosome problem. Stay tuned.
Yep, great. And with the 45 seconds that we have left, since this is an investor conference too, I want to be sure to ask, all of this costs money. Where is Capricor's current financial status, and how long will that fund the company?
Yeah, so we have funding, let's call it through the end of 2017, and that can allow us to move forward with the things that we have already taken on. Obviously, we want to move quickly with the registration trial in Duchenne muscular dystrophy. We certainly are going to have to think about how we're going to do that. And, we're a small biotechnology company. Money is what we burn and what we need.
Excellent. Well, Linda, again, most sincere congratulations on the data this week. I think that was really important for the field, and thank you all very much for attending.
Thank you.